let me see it because I'm waiting for it to come up. Okay, perfect. You are live. يعطيك العافية.
Okay, شكرا. Thank you. Bye. الصبر يطلع ما اعرفش. Okay, good evening, everybody. I would like to thank the hosts for inviting me to give this lecture tonight. My name is Ali Fattom. I'm from Nahaf, North Galilee, Palestine, and I am the Senior Vice President of the Research and Development at a small company next to the University of Michigan. It's called Blurwillow, and I am an adjunct professor at the University of Michigan. And tonight we will be talking about COVID-19 and vaccine development for COVID-19. And I would like to first give you an overview, and then we'll give you some approaches for vaccine development and our work on the vaccine development at Blurwillow Biologics in Ann Arbor, Michigan. If we look at the COVID-19 virus and we compare that to the SARS virus that happened in 2002 and 2003, we see that there's some resemblance between the two. They both belong to the same family, the SARS viruses. And they are RNA viruses, which means a lot of changes can happen in those. But there are big differences between the old SARS and the new SARS. The first SARS, or the old SARS, was really caused an epidemic in some places, some areas. It became infectious, and it disappeared after 2 years. So that limits our ability to look into what kind of vaccine we want to develop for this virus, and therefore we're not able to test vaccines. The new SARS, the COVID-19 virus, is much fit compared to the old SARS. It's caused lots of disease, and now it's a pandemic, meaning that it's really occurring all over the world without restriction to some area only. So if we look at the fitness of the viruses, we are comparing the attachment to receptors. This virus attach itself to the ACE2 receptor on the cells, on the human cells. And when it attached to it, it causes the enzymes inside the cells or to activate the virus and help it in coming in so it can propagate and can give more viruses, and it can infect more and more cells. So the ACE2, which is the receptor for both SARS, is in humans and also in bats, which means, or we believe that this related to the bats, and it came from the source from the bats. And of course, the antibodies that are generated by the SARS, the old SARS, can neutralize the new SARS, but less effective than the old one. And to confirm that we are looking at the same binding, we can say that the SARS, the new SARS, and the old SARS, they can bind at different rates to the bats and to the human cells, which means that their source could come from the same area. And when you look at the infection, when it started at Wuhan in China, we can see that most of the infections early on were related to the wet market, where those organisms or these viruses were identified. Sorry, and most of the infections were related to the wet market, But with time, what we've seen is that it starts to spread, and therefore the spreaded virus is really a little bit more fit than the original virus isolated from the cases related to the wet market. And this is new because the fitness and the ability to spread all over, it becomes really more and more obvious compared to the old SARS, where we had some kind of restriction there. So the virus is really changes, and the change in the fitness is really coming from a small change in the RNA of this virus. And we can see that how this virus really is spread. When we look, and now just a comment here is that we are learning every day, we're learning something new about this virus. It's not something typical, and every day there is new publication, and even the New England Journal of Medicine is updating every day about what's going on in the field. So if you look at the number of the viruses in the nose and in the throat of people, we can see that for a few days, the virus will propagate in the nose and in the throat, and even asymptomatic people are shedding the virus. So we have the virus in our nose, it grows, propagate, and we're spreading it around without knowing that we are sick. And with time, that will become symptomatic, and the disease starts, the COVID-19 disease starts. Now, the people at risk here are mostly people with pre-existing conditions or immunocompromised. Those could be the elderly with of chronic diseases like diabetes, like heart disease, high blood pressure, and so forth. Those are very high risk. In addition to that, I believe there is something else here that the immune system, when you become elderly, the immune system is not as fit as it used to be, and therefore the response to the immune response to the viral infection is becoming more and more serious, and therefore the immune response becomes weak with time, and therefore we cannot really fight the viruses the way we used to fight it when we were young. That does not mean if you are young and you have some pre-existing conditions that you are not at risk for this virus, or the virus can be fatal. The other point that we would like to do here is that the elderly, the target people, or the more and more people are getting infected and dying because of that. And if we look at the distribution, we see that more and more people in elderly homes are getting sick. So therefore, we need to look at ways of preventing this disease. And one of the issues that, or one of the major component of preventing the disease, not only the immune response, and we'll talk about in a minute, but also how can we prevent the transmission of the virus from nurses, from visitors to these elderly in these homes. So there are several ways to tackle this virus. There is a treatment, which is to prevent the virus from existing in the nose and propagating there. There are antivirals, there are antibodies, there are non-specific treatment and existing, like looking at existing drugs. To antiseptic, we at Blurwillow Biologics, we just came up with a nanoemulsion. It's an oil and water nanoemulsion, which has detergent and has surfactant. And we can put that in the nose. It swabs, and you can swab the nose, and that can exist there for about a couple of hours. And therefore, if a virus comes in, if a virus is existing, we can clean the virus, and we can prevent it from to exist there. That's the idea behind the nasal swabs. And we know that this specific nanoemulsion could kill the coronaviruses, and also can kill the influenza viruses. And also we have some evidence that also the COVID-19 could be killed by this virus, by this nanoemulsion, sorry. So how the virus works, and and is very important to know, because if you know how the virus works, then you can tackle this virus. One of the ways is to look for antivirals. The antivirals, there are some antivirals that were developed for Ebola virus by Gilead, which is the remdesivir, and that can be used, and it's been under clinical discussion right now. But still, if you look at the other ones, is really trying to repurpose some existing medicine, like a chloroquine that everybody now talking about, and the hydroxychloroquine. And the hydroxychloroquine and the chloroquine, they are working at different levels. One of the one of the levels is that there is antiviral activity by the chloroquine in the cells that are infected with the COVID-19 virus, the SARS-CoV-2, and by blocking the transcription, the translation, and also by the propagation of the RNA in the virus, and also the assembly and the budding out from the cells. So the virus can be blocked by means of the chloroquine. People talk about the pH, that changing in the pH will change the dynamics of how the virus is propagated in the cells, and therefore could be the antiviral activity with the chloroquine can be useful. The other main area is that the chloroquine can block what we call the cytokine storm. The cytokine storm is at one position, stage of the infection. There is a lot of cells and like neutrophils and white blood cells producing what we call cytokines, like hormones excreted by these cells. Those hormones or those cytokines can, in fact, cause a whole kind of storm that would block. Would cause a lot of damage to the tissues, to the systems, the kidneys, the heart, the lungs, and therefore, what this chloroquine can do is to block that storm and prevent that activity. Sorry, prevent that activity of the cytokines, especially IL-6. And can block the activity of those. So therefore, chloroquine is a good approach. Now, there are some tricks here now that we have to work on how this chloroquine, which is a malaria's vaccine against malaria, and in a different kind of population, how we are going to apply it to people who are sick with COVID-19 or infected with the SARS-2. And therefore, we are required to do some clinical trials in order to evaluate how this thing can work, what would be the dose that we have to give it, at what stage of the infection or stage of the disease this can work. Because if you are a COVID-19 patient and you are sick, the dynamics and the internal hemostats is different than the normal people, and therefore the dose that will apply in one place cannot be good in the other place, and there are some toxicities that we have to take into account. And therefore, although we've seen indication and anecdotes that the use of chloroquine and hydroxychloroquine can help in working with the COVID-19 and also help patients to recover. But we don't have a placebo-controlled clinical trials and that showed without any doubt that chloroquine works this way and can give you whatever you are looking for. So the conclusions from all these studies are not there yet, and there are some studies that are ongoing right now with the chloroquine and also with other repurposing. The other antiviral is the remdesivir, and it's still in clinical trials. There are anecdotes. It's been used under compassionate use in different places, but definitely There are some leaks lately from the University of Chicago that they are showing that it works and so forth, but we are still awaiting the outcomes from the placebo-controlled clinical trials that are ongoing in different places in the world. Now, just to go back and look at the virus, the best thing that humans have ever introduced are the vaccines, creating a vaccine that can prevent the disease, which is really a very important approach. Now, if you want to create a vaccine, you have to look at the virus and understand exactly how the virus is built and really what are the stages and where you want to intervene. Most of the viruses, we would like to intervene at the first step, which is the binding of the virus to the cells between the virus and the receptor on the cells. If we are blocking the virus there, then we are able to mediate neutralization of the virus and prevention of the disease. So if we look at the SARS-2 or the COVID-19, we know there are these spike protein, the crowns on the surface of the virus, and those crowns are comprised of what we call a spike protein. It has two components: a spike 2, which is the binding, the internal side bind to the membrane of the envelope of the virus, and there are a S1. S1 is a trimer, which means that our three molecules are sitting like this. And those three, each one has at the edge what we call the receptor binding domain, RBDs. Those three, they are the pre-fusion when it binds, and then the virus can internalize. So if we are able to block this spike protein by having antibodies to the RBD or the receptor binding protein, then we are in good shape. We can really block the virus. And most of the people and most of the vaccine I'll be talking about in a minute are really targeting the spike protein, whether pre-fusion, post-fusion, a portion of that, the RBD only. And that's to prevent the binding of the virus to the ACE2 in the lungs and in the nose and in the throat. So that's the target for the vaccine development. Now, if we look at the different approaches to this, and there are different technologies been employed for the development of vaccines. At this point of time, in today, which is April 19th, there's about 70 different companies and institutions trying to develop a vaccine. Everybody wants to do a vaccine, and that's great because the chances for success of vaccine is in the teens in normal cases. But under the circumstance, maybe we are really rushing, but it's an emergency. So there are vaccines that are based on DNA or RNA. These are the nucleic acids, and these are injected into the people, and the body or the cells of the host will translate and will transcript and produce the RBD or the spike protein internally. So you are injecting, we are making the antigen, and then our immune system will respond to that antigen. This technology advantage is that it's fast, and the speed by which we can get there. So, and it's synthetic, so there is no need for big GMP manufacturing and so forth. It's really straightforward. Now, the only trick here is that these technologies are not known for successful track record somewhere else. So basically, we have new technologies. We don't know whether it's going to work or not. There is some indication in the animals, but animal data, it can also mislead. And in our jargon, people who are in this field of vaccinology and get bitten more than once with animal models, is that the animals are stupid and they can lie. And therefore, let's be careful about really how much we can expect. There are also inactivated virus, the old way: isolate, identify, inactivate, re-inject, and create an immune response. There is also live attenuated, which is taking the virus, the same coronavirus, and attenuate, make it less virulent or non-virulent, and inject it, and then you can make antibodies to that. There are also another replicate vectors, which is you take a virus which is not harmful, and you inject the gene into that virus for the spike protein. So you have a virus, non-harmful virus with the spike protein from the corona, and therefore you give it to the host, to the human, and then you can get an immune response to that. There are mainly many others that are looking at the subunit, meaning take the virus, you grow the virus in cells, isolate the spike protein, and purify it, and then use that as your vaccine. Now, the good thing about this approach, which is the classical approach too, is that you can put adjuvant, which are enhancer to the immune response. When you enhance the immune response, then you can get different parts of the immune response, immune system activated. And you go ahead, and there are other technologies, but those are the main ones. And right now in the clinic, we have about 6 different vaccines. We have RNA vaccine, we have a virus, attenuated virus, and we have a vector, mostly vectors. The attenuated is not there yet. And we are working on different approach. Our approach is this, that when you look at the flu vaccine, where you have a matching virus, a matching vaccine, 100%, the efficacy in the last 15 years did not exceed the 40%. We actually achieved 50% 1 year out of the 15 years. So you have the great virus, the great vaccine for the influenza virus, and your efficacy is less than 50%. And that's really a problem. Now, that does not mean, so I don't want anybody to misunderstand me, I'm not saying that the flu vaccine is bad. Even if the efficacy is only 20% or 40%, your symptoms can be also alleviated by the vaccine. So basically, the vaccine can help in the outcomes of the infection. The problem there is that the virus can still infect. The virus can still exist in the nose and in the throat and in the lungs. It takes time to recruit the immune system, internal immune system, to recruit it to the lungs in order to fight the immune response, the viral infection, I'm sorry. So that's one. Two is that none of the vaccines given injectably or by a syringe in the arm can prevent colonization and transmission. So we have a problem here that the transmission part of the process of infection is almost independent of the immune system. The immune system can prevent the infection, but when you get that immune system through injectable, you will not be affect much the colonization and the spread. And as you can see, the virus spreads all over the world. So you want to have some kind of technology that can block the virus and prevent it from infection in the nose and propagation in the nose and the throat before it gets into the lungs and cause the disease. So that's where we come into the game here with a technology that was developed at the University of Michigan. And this technology is an oil and water nano-emulsion, a small droplets, about 400 nanometer, that can be mixed with the vaccine, which in this case could be the subunit, and then you give it in the nose. When you give the vaccine in the nose, it will be processed locally, and then it will induce systemic immunity, similar to what is happening with the injectable, but some of the immunity remains locally, meaning that you are recruiting cells and the cells sit in the nose or in the mucosa. And therefore, when a virus comes from the outside and that virus try to infect or to bind to the cells in the mucosa, then the local immunity comes first. This is what we call the wave one. And then once you tackle the virus there, You are waiting for also the wave 2 comes, and then you can clear the virus before it comes in and cause disease. So that's the approach that we have, and I don't believe that any of these 60 or 70 vaccine candidate out there can give you this kind of immunity, and therefore we think that it's unique to us that we do that. Now, is that really what is happening? Can that work? Well, as again, to as much as you can see with regards to the in infection and colonization and how much you can believe in animals. We looked in the RSV, the different virus that cause disease in elderly and also in young infants. And we immunized animals. In this case, we immunized monkeys in the nose and in the injectable, same vaccine. Went into a group that received it in the arm and a group that received that in the nose. And we challenged these animals with the virus. What we've seen is that the animals that received the vaccine in the arm, intramuscularly, generated immunity, and that immunity also was able to prevent the virus from existing in the nose and in the lungs for only 3 days. After 3 days, the virus broke through and start propagating in the nose. So there was not ability to prevent the colonization in the nose. While the animals that received the vaccine in the nose were clearing the virus within 1 day and did not come back ever, not into the lungs and not into the nose. That gives us an indication that what we call mucosal immunity, the local immunity produced and existing in the nose and in the mucosa, then this can play a major role in having the wave 1 and the first line of defense against these viruses. And therefore, if you don't have it, then you will have an issue because the virus can exist in your nose, can propagate in your nose. You can transmit it without knowing. You are not sick, but you are carrying the virus. And therefore, being able to produce or to develop a vaccine to prevent colonization is a very important part of our work. And again, we are targeting the trimer, which is the pre-fusion, which we think it can be more effective than the post-fusion or the RBD alone. And we are in the middle of getting that material. Now, why we think we can, when we think we can have a vaccine? And that's a question that everybody is asking. Can we have a vaccine really tomorrow? Can we have vaccine in 3 months? Now, we have to take things rationally and reasonably and look at the vaccine development in the art. Vaccine development on normal days and normal times, it takes about 5 to 10 years. And the success rate is about 10 to 20% at best case, meaning that you can start with 100 vaccines, you end up maybe with 1 or 2 that will reach the endpoint. And therefore, it takes you a sequence, preclinical, phase 1, phase 2, phase 3, registration, commercialization. Now, if you look what happened with COVID-19, the FDA and the EMA in Europe and about 20 different countries with different regulatory groups came together with the WHO and came up with a recommendation. It's called emergency use authorization, which means that they will cut off some of the animal studies. You don't need to do the preclinical everything. Show me that your vaccine creates antibodies that are useful. Then I will allow you to go to toxicology study fast, and then you get into the clinic. You can do phase 1 instead of following the normal conditions. You followed your volunteers for safety for 6 months or 12 months. That's fine. Now you just follow them for 2 weeks or 4 weeks after the last immunization. If you immunize twice at 0 and 4 weeks, then you follow them for 8 weeks and get the data. Is if you are safe, if you are immunogenic, you are moved into phase 2. In phase 2, there is a new design. Phase 2 is not anymore to look for dose and to look for target population and so forth. You just go in with immunization according to what you've seen with the safety, and then you follow the infection rate. So you have placebo controlled, and you can say a priori beforehand, I will be looking at 20, I will look at 40 cases and 60-day cases, and then I will look what is the efficacy of my vaccine. If I have a good vaccine, the majority will be in the placebo, and I hope only few in the vaccine. If I reach significant protection at any point of time of this clinical trial, it can go up to, let's say, 6 months. And in 6 months, I see those cases, and I see efficacy, I go back to the FDA or the EMEA in Europe or any other regulatory, and I will start actually manufacturing, and they will give me the emergency use authorization to take this vaccine and start commercializing it. And therefore, when you ask people today, how long is it going to take to make a vaccine, I would say 12 to 18 months. And because it depends on where you do your clinical trial, how many people you are going to recruit, and all that, and manufacturing, and all the aspects of that. And also the FDA, even though is giving you this margin, a very nice margin to develop it fast under emergency conditions, but still the FDA is responsible for the safety of these products, and they have to know that this is a safe product, especially that you are going to give it to so many people, and you will never know what is going on with that. Now, one point that I forgot to mention, with COVID-19, there is an issue what we called disease enhancement, and that disease enhancement can happen with the antibodies. Meaning you are generating a vaccine and you give it to a person and they generate antibody. Those antibody can enhance the disease, not protect against the disease. And that happened in history several times. For instance, the RSV vaccine in 1962, the first vaccine that they did for the RSV, kids that received the vaccine and the next season got exposed to the virus, we had 2 deaths there. That's called disease enhancement. And you want to make sure that your vaccine is not disease enhancement or cause disease enhancement or hyperinflammation. We call it type 2 inflammation. You need to have a vaccine that will produce a protective immune response and safe immune response. And those are the things that the FDA is looking at. And those are the things that we'll be looking at when you go into your phase 1 and into your phase 2. Therefore, the development of the vaccine, in order to get to a clinical trial, you have to do, in our case, we are working on this prefusion S protein, a spike protein. It takes about 2 to 3 months really to get some animal data, and then you have to go to a toxicology study because it's intranasal. We have done toxicology studies before with other vaccines, but you have to do it for this one too. Once you have that, you are open to get into the phase 1 clinical trial and to be followed by phase 2. So in our case, my expectation is if we are starting today, we should have a vaccine in 12 to 16 months. And therefore, I would not really make it a magic. It's going to take 12 to 18 months to see the first vaccine ever if we are successful and generating the right immune response. So in summary, there is some light at the end of the tunnel. We want to develop drugs or medicines that can cure the disease, and that should give us some margin and breathing space in order to develop the vaccine. We cannot just wait for the vaccine without doing any work on the drugs. But the drugs will not really come in place of the vaccine. So some people think, okay, we have the remdesivir and we have also the chloroquine. Who needs vaccines? No, the vaccines are important because they are preventive, meaning that you are getting it in order to prevent the disease, prevent the infection. And therefore, I think that what we are seeing with with those drugs or with those medicines will give us a breathing time, and also will maybe allowing us to get out of the house and join our, back to our lives, not as normal as it used to be. First thing everybody will do will be cut your hair or get a haircut, and I need one too. So with this, I will stop here, and if there are any questions, let me know. Thank you. Okay, I see that there are no questions. So if you have a question and you want to, I just don't see the, hello? I cannot see the questions. Okay, okay, I have my wife's telephone. She has the. Okay, I have my wife's telephone. She has the. I have my wife's telephone. She has the. Okay, so does hot water and salt help prevent the virus from entering the lungs? No, I cannot use this one. Well, you can have salt and water. You can have anything to try to remedy some of the symptoms. But definitely, I am not sure about the salt and water that many people are talking about. Some people talk about herb medicine. Anything that can help if you are really sick, and you take it. But there is no evidence, scientific evidence, that salt and water would help or would kill the virus. Of course, but you cannot reach the cells because the virus has been produced, and then you kill maybe some, but I'm not sure about that. The other question is that any indication that use of NSAIDs has any adverse effect on those who have COVID. And I think that people were talking about ibuprofen and other medicines. There's some maybe anecdote, but there is no evidence that you cannot use these things. So, but I would leave it for a real doctor to talk about it. And if you have a question really like this, it should go to the doctor, physician that knows about these things better than I do. An indication that use of so do you believe that doc is in use should trying a ozonation of the blood? I don't know what is that, but definitely I remember that during the septic shock arena, people were talking about passing the blood through a column so you can get all the endotoxin or the LPS out of the blood. I have no idea. Even if the virus is still in the lungs and the virus is still in the cells. So even if you clean the blood from the virus, would that really prevent the disease and clear the virus? I'm not sure that would be a good case. Any idea whether or not those who have recovered from immunity, any idea whether the vaccine will provide lifelong immunity and if booster will be needed? That's a good question. That's the question that I like to answer. People get, viruses in general. When you get a viral infection and you recover from the virus, you will be gaining immunity. That immunity could be antibodies, can be T-cell mediated immunity. And also if the virus in the nose, you can get mucosal immunity from that. And that's the best immunity that one can get. So for normal viruses, okay, and now we are looking at what's happening with the COVID-19 virus. COVID-19 virus, people are looking into this. Some people are recovering without antibodies. Some people recover with antibodies. So some people recover with both antibodies and T-cell mediated immunity. But we are seeing lately that in some cases, like in Korea, that people who are recovered from the infection, they are getting in second time infection. Now, is that because first they were released based on not good tests, and therefore they did not clear the virus from in the first place, therefore they got them back, or they never had an immune response? Or if they had an immune response, you don't know the quality of that immune response. So therefore, what I would say is that, yes, recovering from the virus is a good immunity would be there, and that would be a long term. Now, for the vaccine, the best vaccine is a vaccine that can give you immune response that's similar or close to the infection. That's what we know. Convalescence from infection is the best antibody, the best immunity. Therefore, I believe that the vaccine should give you a good immune response and a memory. A good vaccine should create memory that can be triggered upon re-exposure. And this is where what I was talking about, mucosal immunity versus systemic or internal immunity. Systemic immunity, there is memory, but in order to trigger that memory, the virus has to sit, and the system has to recognize the antigen or the specific antigen on the virus. So there is a lag time between the infection or the exposure and the triggering of the immune response. And therefore, that's really can be an issue. This is why I was talking about mucosal immunity you don't need the virus to sit and to expose a threshold of exposure because the immunity is localized. And that's really the difference. Now, do we need a booster? Possibly, maybe in years. It depends on the quality of the vaccine and the quality of the immune response. And as I said, which adjuvant you are going to use would make a big difference in what arms of the immune system you are going to be creating and how strong is that. Now, boosters, we know vaccines can boost. A good vaccine will establish good memory, and that good memory can be triggered by either vaccination or exposure. Sometimes this memory can trigger non-specifically, meaning that we are exposed to an antigen which is non-related but cause all kinds of immune stimulation, and therefore the memory can be called in. Where we can get the oil nose swab that you mentioned. This swab is going to be on the market in 2 weeks, and it's going to be on Amazon and going to be probably in CVS. So I'm not in the process of making any advertisement, but this will be available and on Amazon, I believe, within a week or two. If someone had a minor symptom such as loss of sense of smell and taste, would they be able to get a good immune response enough to be detected by IgM and IgG test? Well, it's an exposure. An exposure, remember that the immune responses, you have to process the antigen. If you don't have enough antigen, you might not have a good immune response. So therefore, it depends what is this loss of smell indicate, whether you have enough virus or you don't have enough virus. But possible, yes, you are going to get IgG, and also you're going to get mucosal immunity if you have it in your nose, and that's very good. Subclinical exposure is a good way of inducing immune response and immune memory, and therefore, yes, I believe that would be. And the IgM is not a very good, but it's really, and it's transient. The most important thing is the IgG and also the IgA in the nose and in the mucosa. Both of them are very important and can be triggered by exposure to the virus. Any indication that COVID-19 can mutate or evolve, what would that complicate vaccine development? You said wet market versions of virus were less robust than current virus. Yeah, what I said is that in the beginning there was some adaptation, although the sequences did not change for the virus. So the RNA and the spike protein, if you look at the different, right now we are sequencing, and people and labs are sequencing every isolate from every country. We don't see huge changes. The RBD is still in good shape. All these RBDs in different places, they look similar. So therefore, the immune response will be good for all of these isolates. Now, if there is a change, and I mentioned in the beginning about SARS-1 and SARS-2. SARS-1, if we had the vaccine from SARS-1 applied to this, we could have antibodies that are cross-reactive and can neutralize the SARS-2. We've seen that if you take animals with the SARS-2, SARS-1 antibodies, you can neutralize the SARS-2, but not as efficient. And you are talking about a 75% homology. And so a mutation and changes that are happening is not going to be like the flu virus. This seems to be like a steady, and hopefully that's not going to change on us. But knowing if we develop a successful vaccine and there is an emergence of a new one or a change in a new one, we can apply the same technology with no need to do all the other things. Just apply the same technology, produce the vaccine, and you have some assurances that this would be useful, because you learn so much from the current one about what kind of immunity, what are the outcomes, what are the endpoints that you want to look at, how the vaccine would look like. All these questions will be in place. So when you come to produce a vaccine against a new emergent of the same vaccine, of the same virus, then that's going to be a very good value. So developing the current vaccine is going to be a learning for us, and that learning would help us in combating a new area. If vaccine development is successful, could we eradicate the disease like smallpox? We wish. Of course, you know, we are trying to eradicate the polio for many, many years. There was never, ever, you know, a full vaccination schemes. There are always barriers to vaccination in poor countries, you know, the cost and so forth. Now, WHO is doing much of this work by trying to reach every place. And I think it's very ambitious to say that we're going to eradicate. We're not going to eradicate, but we can contain, we can prevent, we can make it really a small occurrences here and there. Polio vaccine, we thought in 1990s that we eradicate like the smallpox. Smallpox, we eradicated it. There is no smallpox on the face of the earth, but in some laboratories here and there that they are keeping some stocks. Now, the justification for keeping that is saying that in case something happened, we have the strains that we can do vaccines and stuff like that. God knows. But for the polio, again, we still have polio. We still have polio in different areas. And people talk about barriers and, you know, like, can you reach every person on the face of the earth and immunize? No. Can every country really look at this as a vaccine that's going to give it to everybody? We don't know. And so therefore, I would not say eradicate. I would say prevent. I say contain. I say, you know, reduce it to a maximum. That's the way that we are going to look at. How reliable is serology testing? We are reading that they have low sensitivity. Yes. There are many false negatives and false positives. There may be sometimes non-specific binding that you don't know, and maybe there's some cross-reactive antibody that has nothing to do with the exposure to the virus. I think that they are using the serology now, which is the IgG and maybe IgM also, although it's short-living immunoglobulin. They are looking at it to test for humoral immunity. So the idea is that if you have so many people with antibodies, meaning that you have immune response and you can count on herd immunity. Now, the only problem that I see with that is that the antibodies alone is not enough. You need to have other arms of the immune system. But it's a good sign that if you have antibodies, meaning you were exposed, you generated an immune response, therefore we assume or presume that you will be protected against infection, and therefore you can go back to the field to work and to enjoy yourself outside. But I think that we have always to be careful. These things should happen gradually, should be happen under control conditions, and we should not really forget all the precautions that we took on daily basis and before we go back to normal life. Our life after COVID-19 or SARS-CoV-2 and after is going to be different. There must be a change in behavior, a change in how we handle our washing hands, staying distance, social distance. All these things is not going to be going back to hugging and kissing. It's going to take some time until we get there. So I think that don't be that optimistic. It's going to take time. Okay, I think that I got all the questions. So Khalid is, yes. What's the name of the oil nose swab in Amazon? Just to help us search. It's going to be called nanobio protect, which is the nano emulsion for swabbing the nose. The indication is that you are, it's called skin sanitizer, and also the nose, where in the part of the nose is also skin, and therefore it's indicated there. So it's not a prevention of colonization. We cannot make that a claim. You need clinical trials for that. I missed the first part when you mentioned the name of the oil. We'll try to order for my customers. I work for CVS and will make sure to get the store soon. I think we are in discussion with CVS. I'm not sure where we're at, but the corporate CVS are in discussion with the Blurwillow for nanobio protect. And I think that the first place to come out is going to be on Amazon. Will a successful vaccine against COVID help counter anti-vaxxers propaganda and help against misinformation and stigma that some conspiracy theorists promote? Well, the anti-vaxxers, they are really fighting against any vaccine. So even vaccine that showed that they saved lives for many years, those guys are sometimes it's really impossible to convince, and they are making a lots of damage. And we are talking about vaccines like measles. And if you guys in Michigan or other places, you heard about the measles coming back and the spread, and that's because people said, you know, if I don't have measles, I didn't see measles, you know, in the last 25 years or 30 years. So why I would immunize my kids and my grandkids with measles? Now, you did not see it because we have a herd immunity, and that herd immunity because the others were immunized, therefore they are not transmitting or and spreading the virus to your kid, and there your kid did not get infection. Now, if many of the kids will not get the virus, you are not going to have that herd immunity, and therefore you are going to have outbreaks, and that becomes a major problem. Now, are we going to be able to use any logic with these guys? I don't believe so. I think that in other countries like ours, in Palestine and in Jordan and in the Arab countries where we are not as rich, the immunization rates is about over 99%. So basically, this kind of, I would look at it as a luxury, whether intellectual, whether cultural, whatever you want to call it. That anti-vaxxer thing is really a luxury that we cannot afford. We have to fight it, and we have to make sure that these things, are not going to be stopping people from getting the vaccine. Otherwise, outbreaks and pandemic and epidemics is going to happen here and there with serious, very serious infections. Okay, I believe that, no? All right, thank you very much, and I appreciate the time, and I hope that everybody is safe and that we will have something soon for everybody, and if we are successful, then we all are going to celebrate. Thank you very much. Appreciate the time. Good night.